And here we are again, the daily highlights of Falling Walls and Berlin Science Week with just a short edition today. Great to have you joining us. Welcome. My name is Astrid Frohloff. I'm a German TV moderator and journalist, and I have the pleasure to lead you through this highlight hour. This session will give us a short overview about great events coming up in the next 24 hours before we will start our daily winner sessions. Every day we will celebrate some of the best science has to offer in 2020, and we will experience the breakthrough of the year across 10 disciplines. Distinguished juries will join us, and our first category today is life science. And I'm now connected virtually with the jury chair of this category, Marja Makarov, Director of Biocenter Finland. It's great to have you with us. Hello, how are you?
Thank you. Hello.
Mrs. Makarov, could you describe what was actually your job leading the jury for this category, life sciences?
Thank you. Yes, indeed. So the job of the chair is to lead the evaluation work by drawing from the specific expertise areas of each of the jury members and then amalgamating these knowledge areas into high collective intelligence.
Mm-hmm. Can you give us a sneak peek into the upcoming winner sessions? What were the hot topics? What are the issues?
Yes, indeed. We had 64 proposals all in all, and those hot topics could be perhaps listed as follows. So, the first category, new antibiotics, new vaccines, new drugs, then brain research and therapies for brain cancer, gut microbiology, medical technology, plant biotechnology, and evidently coronavirus and COVID-19 related topics.
I guessed it was quite difficult to find out the winners. What were actually the criteria for finding the most innovative contributions?
Well, the criteria were extremely demanding. First of all, excellent science, but this was not enough because the research findings had to be translated into innovations that benefit society. Societal impact was sought for. And it's, in the best cases, the innovations were groundbreaking, meaning that they were something totally new, opening up new avenues.
Do you have maybe a recent favorite example of a real breakthrough in your field?
Well, I would like to mention the gene scissors, the CRISPR-Cas9 technology, which will be awarded the Nobel Prize on the 8th of December. And this is absolutely groundbreaking science, paradigm-shifting science, and then innovations which will impact us from public health to plant biotechnology.
So let's see what kind of breakthroughs we will find during the winner sessions today. Thank you so much for the moment, Mrs. Makarov. We will talk to you later. Thanks a lot.
Bye.
Well, there is one field we desperately need groundbreaking breakthrough innovation, and that's the necessary shift towards a sustainable economy. And this is the field of scientist Emily Schackberg, the director of Cambridge Zero, the University of Cambridge's climate initiative. And we are very glad that she is contributing to the Berlin Science Week. And I have the chance to talk to her now. I'm virtually connected to her and talk to her about her vision of zero carbon future. Thank you for joining us, Emily. It's a pleasure to have you with us.
Oh, it's a pleasure as well.
The climate crisis is, next to COVID-19, the most discussed topic of our time. What aspects of this problem would you wish should get more coverage?
Well, I spent much of my career working as a polar scientist, and I would say that having wider understanding of the scale of the changes that we're seeing today in the polar regions would be really welcome. In the Arctic, the scale of the sea ice retreat is almost unimaginable. At the end of the melt season, we're seeing a loss of sea ice compared to 40 years ago that's equivalent to the area of the United Kingdom, France, Spain, Germany, Poland, all put together. In the Antarctic, the vast West Antarctic ice sheet is melting, and we are concerned that indeed it might be an irreversible possible retreat. And if the ice sheets over Greenland and West Antarctica did indeed collapse, that would eventually lead to many meters of sea level rise.
Mm. Well, there are many global, very complex problems which have to be solved in this context. How is your initiative going about fostering the level of international cooperation?
So we're looking very much across all different dimensions of what a university can be delivering. And as you say, looking very much in the context of international cooperation around that. So international cooperation around the science that's required to drive the innovations that will lead us to a zero carbon world, but also cooperation around the education and all the engagements beyond the university itself with business and policymakers.
Okay, what would you say are the top 3 most important scientific advancements we have to get done in order to achieve your goals?
So there are many different areas where there's actually currently today exciting science being undertaken to help drive us towards a zero carbon future. But if I was to pick 3 areas, the first would be in the area of energy technologies, particularly battery storage technologies. We know that so much of our world is going to be transitioned to an electric world, and having that battery storage is critically important to underpin that. The second area that I would point to is transport. It's another major part of our emissions. We know that our ground transport, our cars, are very rapidly moving towards electrification, but aviation is a key part of our transport system, and looking to see how we can deliver zero carbon aviation into the future is a critical research challenge at the moment. And then the final area that I would point to is greenhouse gas removal. If we are to address climate change, we're going to need to find ways of actively removing greenhouse gases from the atmosphere, either by technological means or by natural means, enhancing nature in a way that can help the climate problem. But if we're going to do that, we need to ensure that at the same time, it's also helping the biodiversity crisis that we're also experiencing at the moment.
Well, we're looking forward to meet you again during the session at the summit here. Thank you so much for sharing your insights, Emily Schackberg. Thanks a lot for this talk.
Thank you.
Well, you might have noticed we have got a very dense program for you, full of inspiring insights, deep diving discussions, and amazing workshops. So what is coming up during the next 24 hours? I would like to give you an overview about the program. So one session is Meet the Experts, Fragen und Antworten zu Machine Learning and KI. Also, this is about artificial intelligence and machine learning. This session is going to be in German, as you can see. Another one is called From Atoms to Customers. It's a session by German life science company Bayer. And we have a session which is called Mexican Science Day. Recent results of Mexican and German research in health sciences are presented here. And let's see what else we have. There are more highlights coming up. Let's have a look at the schedule. We have a highlight which is called Una Europa Talk. Resistance is future, you will be digitalized. This is a slightly altered quote from the Star Trek series, as you probably know, in a live conversation from 4 European cities. So the next one is really a good tongue twister. Are we more like we think they are, or are they more like we think we are? This is about science of intelligence. And we have a session called Meet the Prize Winner with the Max Planck Humboldt Research Award winner 2020. And the award winner is Roberto Bonasio. Then we also have something interesting which looks like a lot of fun, and this is our Open Science Slam. This is the last one here on this schedule. The first one on this table is the European Science Diplomacy, Wissenschaft, Politik, Gesellschaft gemeinsam Zukunft gestalten. This is in German. It's an interesting question. How can science, policy, and society manage to create commonly the future and overcome obstacles? And then we have the Citizen Science Digital. This is a follow-up session by AG Weißbuch, a strategy process of citizen science. So I can only encourage you to browse the dense program and add your must-attend sessions to your personal schedule, I think it's really worthwhile to visit a lot of these sessions. So something else which we highly recommend are our daily Falling Walls science circle tables. At the circle tables, world-leading scientists and policymakers from different fields sit together and discuss how we can apply science, research, and innovation to get the world moving again. And tomorrow at 4 p.m. German time, we will discuss an important issue. Let's have a look. It's about combating systematic discrimination in science, and it's about systematic racism in science, a very important issue. And expert commentators and scientists will discuss the transformations we need to correct systematic racism and discrimination in science. This circle is being coordinated by the International Science Council, and obviously this has a very strong political and social impact and is worth to visit. So now to something completely different. We want to throw a spotlight on a more technical issue on mobile health applications. Are they a cure or a blessing? The merger of digitalization and healthcare has been going on for a while, and it's about to get into its mature status. We talked with Claudia Witt. She is a co-director, Digital Society Initiative at University Zurich, about her next steps on this journey and her possible hurdles it provides for healthcare actors. Here is a video.
My name is Claudia Witt. I'm a co-director at the Digital Society Initiative at the University of Zurich, and I'm a researcher at the Charité Universitätsmedizin Berlin. Medicine is becoming more and more digital, and especially Corona has enforced this even more. And one rapidly growing field within medicine are mobile health applications. This includes, for example, all the apps we have on our smartphones, but many other things. And these new developments bring opportunities and challenges. Our event on the Berlin Science Week is a panel discussion, and because this topic of digitalization in medicine is so complex, we really need to partner between the universities, as we have done between the UZH and the Charité. Because of this, this is a joint event. Mobile health applications bring big opportunities. They can support diagnostics, they can facilitate the dialogue between the health professionals and the patients, and they can even foster self-care interventions, but also other interventions. The challenges of this field are multifold. We have to see the bigger picture and to balance between the opportunities and risks. This includes all the regulatory frameworks and innovative creativity. But most important, we have to identify and overcome barriers to digital healthcare so that we develop ways that all of our citizens can benefit and not only those who have already digital competency. With digitalization, our world is already changing, and our contribution here to foster a dialogue, to do joint research, is contributing to a wise shaping of the digital transformation.
Every day we are presenting another short story by German famous photographer Herlinde Köbel. She went out into the field, took pictures of famous scientists all around the world for her new book and exhibition, and my colleague Felix Rundl, Executive Director Falling Walls, talked to her.
Herlinde, we are looking at a photograph of a man smiling into the camera with a drawing on his hand. I think it's waves. This is Karl Dijsselroth. He's one of the top professors at Stanford University and a pioneer in the field of optogenetics. Can you tell us what you found out in your interview with him?
He has a very unusual brain. You know, he can focus and read a whole group of words, a whole block of words, whole pages, and repeat it immediately. And also, he's very unusual also as a researcher. And he always tries to find out where our feelings coming from and also how our brain is working. In private life, he's also unusual because he has 5 children. His wife is also a scientist, and they are taking care of the children by themselves. And he tries to carve out every day an hour to have deep thinking, and he thinks that's very important to be a good scientist.
So stay tuned now as we are continuing immediately with our winner sessions. Our first category today is life sciences. So looking forward to see you again in a minute. Bye. Hello, welcome to the Falling Walls Breakthroughs of the Year 2020, right out of the heart of Berlin, Germany. Great that you're joining us. My name is Astrid Frohloff. I'm a German TV moderator and journalist and have the pleasure to guide you through the winner sessions. You're at the right place here at the winner sessions, where you can learn more about recent breakthroughs in science and society. And over the past months, over 900 nominees from all around the world have made the journey to become the Science Breakthroughs of the Year in their respective category. So in this session, the top 10 winners of this specific category will present their breakthroughs and its profound impact on society and science. And our first category today is life sciences, and I will say, let's start right away. So before we will meet all the winners in this category, life sciences, I would like to introduce to you our guest here in the studio. I'm joined by Dr. Adam Levy.
Hello.
He is my co-moderator. He is a science journalist himself, and he had the pleasure to do interviews with each and every winner of this year. So I'll talk to you later, Adam. I'm also happy to welcome our virtual guest, Marja Makarov, the jury chair of this category, Life Sciences. Nice to have you joining us from Finland, Mrs. Makarov. Let me say that each category is joined by a very prominent jury. All of them are very distinguished experts in their specific field. And actually here you find them on this screen. These are all the jury members in this category, Life Sciences. I was told it was indeed very difficult to find the right decisions and to decide about the winners. So we appreciate very much the contribution they have made here. Let me ask you, Mrs. Makarov, you are a director of Biocenter Finland and you are heading the jury. I'm wondering, after looking through all the proposals for the breakthroughs of the year in life sciences, what are the most interesting hot topics you have found this year?
So let me mention 5 categories. First, brain research and brain cancer therapy. Then we have proposals concerning with new antibiotics and drugs, vaccines against bacteria and viruses. We have medical technology, we have the gut microbiome, and then evidently coronavirus and COVID-19 related topics.
Okay, so if we look at the COVID-19 pandemic and the surge for vaccines, as you mentioned, therapies and public health measures, it seems that the entire field of life sciences is gaining more and more importance in the world. How is your perception of this fact?
Well, worldwide, the coronavirus pandemic has really mobilized the scientific community, and this is what must happen and has happened. Also, the funders have mobilized new resources for this particular area, and I would like to tell you that 1/5 of the 64 proposals that the Life Science Panel had in front of it were related to Corona and COVID-19, and these proposals then dealt with Corona diagnostics, vaccines, drugs for COVID-19, and also management of the outbreak. And there were proposals concerning basic research on the virus itself.
Okay, thank you very much, Maria. For the moment, I'll talk to you later, but let me switch over to Adam. Adam, I was mentioning that you did all the interviews with our winners. How was that experience? I mean, 100 winners we have all in all, right?
I mean, it's incredible to speak to 100 different people, all incredibly passionate about what they're doing, all doing completely groundbreaking work. Yeah, it's been a joy.
Yeah. And we will learn all of them. I'm delighted to announce now our first winner in this category, Life Sciences. And this is Stefan A. Sieber, Technical University, Munich, Germany. He is aiming to break the wall of resistance bacteria. Congratulations. So, Maria, how did the jury decide? What is the explanation to vote for him and his team?
Now, bacteria resistant to antibiotics, this is a daunting problem. And Dr. Sieber has discovered a new type of highly potent antibiotic that appears to elicit no resistance in bacteria. So the jury felt that his research, the novel findings, are critically important for public health.
Okay, so let's have a look at his potential breakthrough of the year.
Breaking the wall of resistant bacteria. Stephen A. Sieber, Technical University of Munich.
We are working in the field of antibiotic research, and we try to find new ways to kill multi-resistant bacteria. One of the limitations of current antibiotics is that they go for a very limited number of cellular targets. And one idea is to overcome this is to look for new, unprecedented modes of action. And that's what we are doing. And we found a small molecule that has at least 2 novel, unprecedented targets, which it addresses simultaneously. And one of these 2, I think, is quite spectacular because it's an enzyme that's sitting in the envelope of the cell, and it's secreting proteins outside of the cell. And we stimulate, so we overactivate this enzyme. And by this, the cell starts to dysregulate its secretion. More proteins are coming out, and some of those start to digest, to lyse the outer membrane of these bacteria. And therefore, they start to bleed out and die. And together with the other mode of action, we think this is something that not only is active against multi-resistant bacteria, and we can also show this, but it decreases the chance of becoming resistant in the future. And we are now working with passion on the further development in a preclinical study.
Can you give a sense of just how big the issue of antibiotic resistance is?
It's a very big issue. It's actually growing. And at the moment, we see with infectious diseases how big the problem can get. And with antibiotic resistance, it's just a slow process. So we have, over the past years, seen multiple antibiotics becoming ineffective. That's the reason, because they have been applied very often to certain diseases. So the bacteria are exposed over a long time to these antibiotics, and they just develop resistance in order not to be killed.
What for you makes your approach special?
If you look at current antibiotics, they target a really limited number of cellular targets. So there is the cell wall, there is the protein and DNA biosynthesis, and then there are a few more. And this is the reason, this limited number of targets, that there are so many resistances, because bacteria have already developed strategies to overcome this. So therefore, we have to look for novel targets outside these classical ones. And there are several hundreds of targets in bacteria that you could exploit. And this is our unique approach, that we look for exactly those that are not yet affected by resistance.
Now, what would stop bacteria then just becoming resistant to this as well?
This is the breakthrough that we have two targets. So this is difficult because a small molecule usually has a certain affinity for one protein, but we have at least two validated. There might be even more, and this makes it harder because then the bacterium has to develop simultaneously resistance to two or even more targets in the cell. And this is the chance of having this simultaneously is much lower. And therefore, we also in the lab see any resistance development, which is a good starting point. It doesn't mean there will be never resistance in the human application, but at least there is less tendency to do so.
Can you give a sense of just how much work it is developing this new compound? How big a team effort it is, for example?
It's a big team effort to develop such a molecule. It has been already a lot of effort. In the previous time, it was a basic research project. There were two PhD students involved that did a lot of synthesis and target validation. And now it becomes even more work because now you have to show it's safe, it's effective, and it's a team of 3 to 4 people working on this at the moment. And of course, they are doing this by passion, very passionate, me too. And we hope with all our dedication to get it fast moving.
So innovation in the field of antibiotics, a very urgent issue. Congratulations here to the winner. And also our next candidate has a very, very urgent issue, a very urgent breakthrough. We are glad to announce the winner. It's Michelle Monnier, Stanford University. She wants to break the wall in brain cancer. And I would like to ask the jury chair, how was it, what was most convincing for the jury to decide on her?
Dr. Monnier's research benefits children and young adults with brain cancer, and it helps understand how the tumors grow and invade the brain. The jury judged the science to be game-changing, a breakthrough in clinical approaches for treating lethal brain tumors.
Okay, so then let's have a look now at our next video.
Breaking the wall of brain cancer. Michelle Monnier, Stanford University.
Among some of the worst human cancers are brain cancers like glioblastoma and diffuse intrinsic pontine glioma. My research discovered that these infiltrated cancers take advantage of growth signals released as a result of normal brain activity. Even more unsettling, we've discovered that the glial cancer cells form synapses, points of direct electrical communication with neurons, and thus structurally and electrically integrate into the neural circuits that they invade.
Can you give a sense of just how different this behavior is to what we see in other cancers affecting other organs?
The nervous system is really important for modulating cancer growth pretty broadly, not just in the brain and spinal cord, but actually in cancers throughout the body. And there's kind of this emerging new field of cancer neuroscience. We really have to understand how the nervous system regulates cancer progression so that we can target not only kind of the intrinsic vulnerabilities of the cancer cells with radiation and chemotherapy, but also disrupt these really powerful signals that are promoting growth and progression.
How challenging is it to study something in living cancers?
One of the huge breakthroughs that has come for brain cancers like the ones that I study and for other cancers have been the development of patient-derived models. And so actually I've developed a number of different cell cultures and in vivo, animal models from the cancer tissue that my patients have donated for research. And so that contribution from patients and using that together with some of the newer tools and systems neuroscience and applying these really powerful neuroscience techniques to study the cancers, we've been able to uncover these really unexpected interactions that I hope we can target for better therapy of these really lethal diseases.
Now, of course, the brain is such an intricate, such a delicate organ of the body. Does that change how we're able to treat cancers that affect it?
One of the important things about discovering this integration of brain cancers into neural circuits is that it is going to enable us to use medicines that we've used for other brain diseases like epilepsy, like migraine, and actually other medicines like those that affect other electrical tissues like cardiology to treat the cancers. The cancer cells we're discovering are really sensitive to these electrical signals. And so medicines we've already designed, to target electrically active tissues can be used in this context. And I think that's a really exciting new direction that I hope will complement our more traditional ways of approaching these cancers.
What is your hope for the future for this understanding and the impact it could have on our treatment of brain cancers?
I think that understanding the growth signals that brain cancers are deriving from the normal brain has been kind of an important missing puzzle piece in developing effective therapeutic strategies. And I think that advances in targeting and understanding the intrinsic vulnerabilities of these cancer cells, in leveraging the power of the immune system to effectively, treat them through immunotherapeutic approaches are going to be really important, but we must also disintegrate the cancer from neural circuits in order to eradicate it. And I think that this line of research is going to be one critical component in successfully curing brain cancer.
Well, Adam, from your perspective, how is this topic connected to other topics?
Well, we're seeing that we're getting more and more tools to investigate the brain in action, and that's affecting our ability to understand the diseased brain, but also the healthy brain, and we'll see that in several other winners sessions we'll have.
So let's introduce our next winner, and I'm glad to announce Haitayu Yang from Shanghai University with a potential breakthrough in COVID-19 inhibitors. And I'm asking the jury chair, what's the explanation of the jury?
Now, Dr. Yang designs molecules that help identify new drugs against COVID-19. We badly need them. So the jury praised the science to be innovative, of outstanding quality, and of course the societal relevance is very, very high.
Okay, so then let's have a look at his potential breakthrough.
Breaking the wall of COVID-19 inhibitors. Haitao Yang, Shanghai Tech University.
COVID-19 is causing a pandemic worldwide, but effective therapeutics remain very limited. In order to discover new antivirals against the SARS-CoV-2, which is the pathogen for COVID-19, we aim at its prime drug targets. First, we determine the 3D structure of main protease of SARS-CoV-2 at atomic level, which is a key enzyme for the viral life cycle. This is also the first protein structure to be determined from this deadly virus. Then we screened over 10,000 old drugs and natural products against this viral target. Finally, we found 2 old drugs, Epsilon and disulfiram, which are strong antiviral drug leads. Now they have been approved by FDA to enter phase 2 clinical trials to treat COVID-19.
How optimistic are you that this could make a difference to the pandemic?
So still this will depend on the clinical trials, because clinical trials are very important. For the phase 1 is to test the safety, but phase 2, to test the efficacy in patients. Because right now, just we have been doing those research work based on the cell-based assays. So those compounds actually can inhibit the replication, the viral replication in the infected cells.
And some of the drugs you found that actually work on the virus, they were invented for things completely different, right?
So they were just old drugs, and some drugs we can see they were just to be in clinical trial to treat the hearing loss. And some are used to treat like alcohol dependence. That's approved drugs. And then we know it can be applied to the COVID-19. So the complete different uses.
Since these are drugs which have been tested for safety before, In theory, how quickly could we get to a stage where we could actually start prescribing them for patients who have COVID-19?
So because right now they have been approved to enter phase 2 clinical trials. So right now, I think just we are still collaborating with the company, and those companies are still collaborating with the hospitals. So I think right now they have initiated this program.
Has this approach of finding a structure and then searching through many existing drugs for something that's active on it, has this been used before for other diseases?
So like maybe just even in HIV drugs research, and also in identification of the HCV compounds and the drugs. This strategy has been used, I think, and successfully been used. And we know just for HIV, right now, just we're using the cocktail and just the strategy to use, different kind of the inhibitors against the HIV protease. And also we know about the HCV inhibitors and the protease, the inhibitors. Actually, so they were very successful. So this strategy has already been used to assist in drug identification, and they have been shown to be very successful.
So that was Haitao Yang from the Shanghai Tech University. And as you can see, our winners are coming from all over the world. And our next winner comes from Israel. I'm happy to announce Eran Elinev, Weizmann Institute for Science. He wants to break the wall of the gut-brain connection in ALS. And I would like to ask the jury, what is your explanation here to vote for him?
Indeed, Dr. Elinev studies the connection between the gut microbiome and the brain. So he has identified in ALS-prone mice gut microbes that secrete a substance that appears to affect the disease. This may open up therapeutic opportunities for ALS, so far an incurable disease. The jury acknowledged the outstanding science that increases our knowledge on the role of gut microbes in physiology.
Mm-hmm. So let's meet him personally.
Breaking the wall of the gut-brain connection in ALS. Eran Elinev, the Weizmann Institute of Science.
In the past decade, we've been one of the first in the world to mechanistically study the microbiome, which is this complex and poorly understood world of microbes that live within the human body from the moment we are born until the moment we die. And we and many others have discovered that these microbes are central contributors to common human diseases, including, all sorts of infectious, metabolic, neoplastic, and even neurodegenerative disease. In the last year, together with a very talented group of students from around the world, I discovered that this microbiome composed of all of these bacteria contributes to the development and the progress of a devastating, cureless, and deadly disease called ALS.
Now, how surprising is this to see the microbes that are living in our gut influencing this neurodegenerative disease?
Despite the many surprises that this revolution entails, thinking that microbes that live in one place would impact brain function miles away from where they actually live seemed to be science fiction just until a couple of years ago. But what we discovered is that some of these bacteria, which only live in the gut, make small molecules which are able to swim into the brain and impact function related to the brain very distantly from where the microbes that actually made them live in.
And now, how is progress going, not just in describing this connection between the gut and ALS, but actually in the search for treatment?
We've complemented the animal model studies with a human study in which we followed a group of ALS patients and compared them to their healthy household controls. And we were astonished to find that both the gut microbes and the small molecules that they secrete were distinctly different between ALS patients and healthy individuals living in the same house. And even when we looked at the specific molecule which we discovered in mice to improve ALS scores, we found that it was missing both in the blood and in the brain of human ALS patients as compared to healthy control individuals. And this raises a huge prospect that perhaps by replenishing the missing microbes or missing molecules, we would be able to help these patients for the first time.
Do you think the gut microbiome has more big surprises like this waiting in store?
Regard the microbiome as a missing organ, which contains an equal number to the cells that our human body possesses and 150 times more genes than our human genes. So it's a huge organ that we didn't take into consideration in 150 years of modern science. And this revolution, which only began a decade ago, is bringing us surprises every single day. And what we're seeing is that almost any physiological function involving the human body also involves contributions by our microbes. And if we would be able to understand the intricate and complex communication channels that take place between us and our microbes, and what happens when they're perturbed and disturbed during susceptibility to disease, I am certain that more treatment options would pop up that involve the microbiome in more common human diseases in the decade to come.
Well, Adam, can you tell us how does this compare to other work in this field?
Well, what we've seen over the last years is more and more work looking at gut microbes and their role in health. But even with this huge body of research, it's incredibly surprising to see the microbes living in our gut influencing something as far away and as complicated as ALS.
Mm-hmm. Yeah, that's true. Well, our next winner is a scientist who is willing to break the wall to next generation biopharmaceuticals. And I'm glad to introduce to you Christian Hackenberger, Leibniz Forschungsinstitut für molekulare Pharmakologie aus Germany. So congratulations, and I'm asking the jury, what do you say, what's your explanation here?
Yes, indeed. Dr. Hackenberger has engineered inhibitors of influenza virus infection, and he has engineered antibodies coupled to drugs which kill cancer cells. The jury thought the core idea of the science is fascinating, the theoretical approach is great, and the breadth of applications is impressive.
So let's get an idea of his work.
Breaking the wall to next-generation biopharmaceuticals. Christian Hackenberger, Leibniz Forschungsinstitut für molekulare Pharmakologie.
Well, we've developed molecules against cancer and viral infections, and we used a very simple idea. We pretty much combined the best out of the world from chemistry and biology. And to develop better and safer cancer therapy, we advanced the concept of antibody-drug conjugates, so which means loading a toxic molecule to a molecule that specifically recognizes only the cancer cell. And thereby we can reduce the common side effects of chemotherapy. And it pretty much works like a Trojan horse strategy. And for viral infections, to treat this, we came up with the concept of a molecular superglue. And with this, we could find a molecule that specifically binds to an influenza virus and thereby making it not infectious anymore. And well, with this idea, we pretty much hope to fight life-threatening diseases. And of course, we are also developing this concept further. Right now, we are working on this to battle against the coronavirus.
And where is this work today? What would it take to get it out of the lab and actually help save patients' lives?
So the concept has very good first results, and we want to try to, of course, bring this further through the clinic. So we are very hopeful to get some really further encouraging results over the next years.
What are your ultimate hopes for what might be able to be achieved by combining, as you put it, the best from biology and the best from chemistry?
To pretty much come up with, in this particular case, to come up with new strategies which are safer and which in the end work better also in clinical models. I mean, we experience in medicinal chemistry that with the, let's say, common strategies that have been pursued over the years, meaning small molecule treatments, that we don't necessarily can target all the diseases that we want. So we need new innovation, new molecules that could pretty much help in the development of new therapeutics there. And when you think about that, a lot of biological molecules, so-called antibodies, work already pretty well for treatment certain diseases. Then thinking about to make this even better with adding chemistry on top to make these molecules more stable or more active would be great. And so therefore we think that bringing together the knowledge from these different areas is really an advancement for the field.
You are looking at what your approach could offer in the fight against the coronavirus pandemic. How much has your work shifted in 2020?
It certainly has shifted a little bit towards this particular target, but we've been working to battle virus infection already before. And so therefore, for us, the next step to apply this now to the coronavirus research was to now use the knowledge that we gathered before and translate it to the next level.
The Falling Walls audience, and as well the Falling Walls speakers, across many different disciplines. Do you have any messages or calls to action that that you can offer from your experience working between chemistry and biology?
Well, first of all, try it. We should not think that as scientists we have, the breakthrough idea in the beginning, which will be the end also the solution. Start doing your research, talk to people, and then along those way, when you pretty much bring in all your knowledge, your creativity, and your enthusiasm for your science, this is when you can really make a breakthrough.
So that was Christian Hackenberger. Adam, it seems that working interdisciplinary plays a very important role for many scientists. Is that correct?
Yeah, certainly not just here, but of course, as mentioned, taking the best from one field and the best from another can lend itself to innovations we just wouldn't see by just sticking within the camp of one discipline.
Yes, right. Okay, let's have a look at our next winner. I am glad to present to you now Sergio Pasca, Stanford University. His project title is Constructing and Deconstructing Functioning Human Brain Circuits. And the question goes to the jury. First of all, what is your explanation, Maya?
Now, a critical barrier to understanding the function of the human brain is the lack of access to the brain tissue. So Dr. Pasca's scientific discovery is that they are able to grow from stem cells in the lab human brain organoids, which can be used for investigation. The jury praised the work, which helps uncovering brain features, normal brain features, and mechanisms of brain diseases.
Okay, so here we go. This is his presentation.
Breaking the wall of neuropsychiatric disease. Sergio Pasca, Stanford University.
We are interested in uncovering the mechanisms of neuropsychiatric disorders. And really the main challenge in doing this has been our inability to directly look and investigate brain cells from patients. And over the years, we have been developing ways of growing in the lab, non-invasively, three-dimensional brain tissue from patients, starting just from skin cells. But we were still not able to study how neurons interact with each other across brain regions and within circuits. So to address this problem, we have recently developed a completely new experimental paradigm called an assembloid, in which we derive different brain regions from stem cells, and then we assemble them so that they actually form circuits. Very recently, we've built the cortical motor circuit by essentially deriving cerebral cortex like tissue, spinal cord, and then separately human muscle, and then put them all together and found that very interestingly, they connect with each other. And you can even control muscle contraction by using various optogenetic and stimulation tools. So we think this is really exciting because it offers access to inaccessible features of the human brain. And more importantly, for our overall goal, to start to understand how dysfunction in the circuit arises in various disorders, including disorders of the cerebral cortex and basal ganglia, but also disorders that affect the spinal cord, such as ALS or SMA.
What lessons have you already learned by being able to grow these tissues in the lab?
I mean, first of all, just even starting to understand how human neurons, what is unique about human neurons. But then, of course, by applying this directly to understanding brain disorders, we've been gaining new insights into how some forms of autism, for instance, or schizophrenia may actually arise.
Similar work has been carried out for other organs as well. But of course, this isn't our livers we're talking about. Our brains are special. Does this raise any ethical questions?
The human brain is unique because obviously it's us, right? We identify ourselves with the human brain. But it's also very, very complex. And the cultures that we and others have been developing are still very simple representations of parts of the brain and not really of the entire brain. Not to mention that they're also disconnected from experiences and sensory input.
Is your goal simply to understand the brain better, or could this one day lead to better treatments for certain conditions?
Well, I'm a physician by training, and my interest is really in understanding brain disorders. That's what motivated most of the work that we've been doing. And it turns out that quite often, some of the aspects that make our human brain unique also makes the human brain susceptible to disease. So I think there's a lot of convergence in terms of just learning about the human brain and intrinsically, learning about human brain disease as well.
So you are at the Falling Walls Winners Sessions. We are presenting the top 10 winners in the category Life Sciences. And we have 4 more winners on our list. I would like to announce the next winner, and this is Giovanni Traverso, Harvard Medical School, and his potential breakthrough is drug delivery and nutrient absorption. So the question for the jury chair is, Maya, what is your explanation for voting for him?
Dr. Traverso has invented synthetic epithelial linings for the gut. And this helps to develop liquid formulations for medication for children. The jury felt that the idea is ingenious. The innovation could transform medication administration to children, which is currently a major challenge.
So let's learn more about this project.
Breaking the wall of drug delivery and nutrient absorption. Giovanni Traverso, Massachusetts Institute of Technology.
One of the key things that we recognize is that children really don't like to take solid tablets or capsules. And so what we set out to do was develop a system that is in a liquid form that makes it easier for the patient to receive their medication. And also, as it's ingested, it can stick to part of the intestine and sit there for a longer period, therefore requiring fewer administrations because the drug has the opportunity to be absorbed.
Now, what will it take to get from where you are now to actually using this with patients as a way to administer drugs?
The main thing is really demonstrating persistent safety as well as efficacy. And so we've certainly demonstrated that already at the level of rodents and even pigs, which really helps us tremendously in advancing the technology towards humans. And so there's further characterization that needs to occur, that is occurring as we speak. And then the initial first in human trials, again, demonstrating the safety, but then also efficacy of the system in enabling drug and nutrient absorption across the GI tract.
And how does this way of administering drugs actually work?
What we developed, we refer to as G-cell or gastrointestinal synthetic epithelial linings. What they are essentially films that form on the inside of the intestine. And on that film, we can include different elements as such. And so we can include medications that can sit in that film and then slowly be absorbed. So that's hopefully gives you a sense of a coating which has medications on it that are slowly being absorbed while that coating is located in the small intestine.
Can you give a sense of just how different a way of administering drugs this is to what exists on the market currently?
The current common alternatives are tablets and capsules. There are certain powders that require reconstitution in water, but those formulations do not provide any enhanced properties such as extended release. As far as we're aware, there are no other examples of using, and this is actually part of getting now of how the system works, is this system actually interacts with enzymes in your intestine that are present there all the time to form these films. And there aren't any other technologies that do that.
Just how much work has it been developing something like this, and does it require collaboration between different disciplines?
Absolutely. And that's a lot of what our team does and has done now for many years, is really bring experts from across different fields and really, brings together the material science aspects of the development of that system. It also brings together the pharmaceutical sciences, as well as the veterinary sciences, as well as medical sciences, all to bear on this major challenge.
What's your ultimate hope for the difference this could make in the administering of medications?
Our hope is really to bring several different drugs now to be administered using this system, particularly for populations that have difficulty in the ingestion of solid material, solid capsules or tablets. So certainly children, but we see significant opportunities where we could help folks who have neurologic disabilities, the elderly, who certainly will need medication for a whole set of conditions, but where oral administration can be challenging because of their health state or disease state, for that matter.
Well, Adam, you are a science journalist. What's the dot about this issue in your eyes?
Well, for me, apart from being an incredibly novel approach, it's applicability to so many different situations, so many different conditions that really stood out for me about this winner.
Mm-hmm. Let's see who our next winner is. I'm glad to introduce to you now Stefan Dübel, Technische Universität Braunschweig. He is hoping to break the wall to corona antibodies. And Maya, please, can you explain us why did you choose Stefan Dübel and his team?
Indeed, he develops human antibodies to cure COVID-19 patients who are expected to respond poorly to vaccination. The jury felt that the research is extremely exciting with immense societal benefit, though still in a preclinical stage.
So let's have a look at his work.
Breaking the wall to corona antibodies. Stefan Dübel, Technische Universität Braunschweig.
Vaccines cannot cure COVID-19. We need an additional drug which can directly neutralize the virus. What we use to generate this drug is exactly the same our body is using, which is an antibody. Antibodies are drugs produced in our own body to fight infectious pathogens. We generated human antibodies out of the human genes, so it's directly the same substance in our bloodstream.
Now, has this approach of using the body's defenses as a drug, has that been used for other diseases before COVID-19?
This is a fairly old approach. It was first tried in 1890 by Emil van Behring, and tens of thousands or hundreds of thousands of children have been cured by such kind of antibodies which are given to you. Today, we are much more advanced in respect of technology producing these antibodies. So Emil van Behring produced his antibodies in horses, but we produce them by technological methods which are 100% identical to the human molecule we have in our blood anyway. So we expect no kind of side effects from this treatment.
Could you give a sense of just how much work it's taken to get to this point you're at today?
It was an incredible amount of initiative from all my people in the lab, all my PhD students and postdocs and technicians. And these are the real heroes of that story because they worked through 3 to 4 months without any break, without a weekend.
How far are we along the process of developing this as a treatment for COVID?
There are many, many groups out there which are today, as we speak for this interview, are the most successful treatments for the disease. So I'm pretty sure that this will certainly help to end the pandemics in the near future. Our own antibody is now already as well in production for the clinical studies.
So it hasn't yet been tested with patients?
It has been tested in the first studies in America, and it was very successful there, protecting more than 80% of the people from being hospitalized after infection, which is, I think, a dramatic improvement of what we had a month ago. So I think this is the way to go, and we hope that our antibody also can contribute a little bit, because you need several different antibodies because the virus mutates, and as far as we know from our experiment at the moment, recognize different mutants in a different way. So we need several different antibodies to treat the entire population.
How far along are things at showing that this could work as a treatment for COVID-19, and what will it take before we actually know that it could work in humans?
I have absolutely no doubt that it will work in humans, because the clinical results from very, very similar molecules in the US have shown that it works. So it's the best we have at the moment. It has clinically shown that it is working to protect people, and so I think there can be no doubt that antibodies can be a very effective measure against the virus.
And we are jumping now from Germany to the United States. I'm happy to announce to you Xiangdong Fu, professor of the University of California, San Diego. He's our next winner in the category Life Sciences, and his future breakthrough is on replenish lost neurons. So Maya, what is the explanation of the jury?
Now, in Parkinson's and Alzheimer's diseases, neurons gradually lose their functions. Dr. Fu has discovered how astrocytes can be converted to functional neurons, and this may lead to reversal of the diseases. The jury felt that the science is absolutely fascinating and the potential impact is very high, though applications are still awaited.
Okay, so let's have a look at his project.
Breaking the wall to replenish lost neurons. Xiangdong Fu, University of California, San Diego.
The breakthrough is to identify a new strategy that converts non-neuronal cells to neurons lost to neurodegenerative disease.
You're looking at replacing damaged or lost neurons, but what kind of disorders is this relevant to?
In fact, we're talking about neurodegeneration as a whole. There are many types of disease, but in this particular case, we used a model of Parkinson's disease, which is characterized by the loss of dopaminergic neurons, a specific type of neurons that secrete dopamine to connect to the motor functions and other neurons.
And how do you actually end up replacing these lost neurons?
Right. So conventional way people thought about this as a cell replacement strategy as a general strategy to deal with this type of disease, not featured cell loss, by using stem cell. But the stem cell has a different set of challenge. Now we just invent a new way to directly convert abundant supporting cell type called astrocyte, is kind of a starfish shape, back into the neuron and then restore those lost circuitries to restore the function.
So you're able to take these cells which are already there and turn them into neurons, but are they actually doing the job of the lost neurons?
Very precise, that we turn the cell back into the neuron in the original locations. So we don't move cell around, just turn switch their identity from non-neuronal cells to neurons.
And where is this work today? This isn't something we've shown in humans yet, of course.
Correct. This is our goal. We try to push that as soon as we can. We demonstrate that on the mouse model, and obviously we need to demonstrate the efficacy in larger animals before we try on humans.
Could you give a sense of how big a challenge it has been to get to the stage you're at today?
The challenge is to actually involve a series of basic research to understand why the cell get reprogrammed from one cellular state to the other, and how you achieve a stable cell line, and how you achieve efficient conversion.
So now that you've actually demonstrated that this does indeed seem to work in mice, does that mean that we don't need to worry about neurodegenerative diseases for humans in a few years' time?
That's our hope, but I don't want to convey such a message that there's no longer neurological disease 5 years from now. And there are many challenges. Mouse has a tiny brain, and the human has a bigger ones. The complexity of the nervous system is much more magnitude more complex than the simple small animals. Thus, we have work to do to improve the efficiencies, to be able to attack neurons, to avoid side effects, so on and so forth, in order to bring precision medicine to individual disease settings or individual patients.
Now we have introduced to you almost all winners in this category, life sciences. One more is left, and I'm delighted to announce to you Holger Puchter, Karlsruhe Institute of Technology, and his project title is Breaking Breeding Barriers by Chromosome Engineering, and I'm asking the jury for the reason to vote for him.
The CRISPR-Cas9 technology is used to edit genes. However, Dr. Puchter has used the technology to engineer chromosomes in plants, and this to provide breeders with crops that are less susceptible, less dependent on pesticides, and are able to cope with climate change. Now, the jury acknowledged the innovative way of taking advantage of the CRISPR technology and the high impact the research and the innovation has on food security.
Thank you. So here comes his presentation.
Breaking the wall to sustainable agriculture. Holger Puchter, Karlsruhe Institute of Technology.
So many people in the world are using molecular scissors to induce changes in single genes by cutting single genes. But what we do now is we go on a totally different level. We don't change single genes. What we do is we are changing chromosomes. For this, you have to know that genes are organized on chromosomes. And this gives us, for the first time, the opportunity to change the combination of genes. So we are not working on single genes anymore. We are working on combination of genes. You can imagine, for example, if you want to have a crop, it should taste well, but it also should be resistant to a certain pathogen. And this you cannot do by simply changing a gene. For this, you have to make new combinations of genes. And this is the breakthrough we achieved for the first time. We are now able to exchange parts of chromosomes in plants. And this is the new thing we achieved.
Now, can you give a sense briefly of how you actually do this rather than just tweaking a single gene? You're tweaking the combination of genes.
We are making new combination, and this means, of course, that it's not enough to make one single cut in the genome, but you have to make several cuts, and then you have to rejoin these cuts in a different way. So in the most simple way, what we do is just we cut two different chromosomes, and then we make new combinations of the respective chromosomes arms, and by this we have some new combinations.
It sounds like you have all sorts of applications in mind, but what would it take to get from where you are today to actually applying this to crops that we're really using out in the field?
The surprising and exciting thing with CRISPR-Cas is that things happen very fast. And I don't want to promise too much, but it looks very promising.
Do you have any sense of when we might be able to expect this in crop plants?
In the very near future, next year.
And how far have you gone to date with this technique?
Well, the latest results we got, the latest exciting results we got, is that we not only are able to change the setup of a chromosome, but that by changing the setup, we are also directly able to change the inheritance of specific genes. So it's one thing to set up these changes. It's a second thing to then influence in a classical breeding process inheritance. And we could show that this also works. So this is a second very important step we just achieved 3 months ago.
And ultimately, what do you hope this technique might allow yourself and others to achieve?
We are all discussing major problems of agriculture at the moment. There are much too much pesticides used. Global warming leads to massive loss of yields in crop plants. And with these technologies, we will be able to produce crop plants that use much less pesticides. And we will be able to produce crop plants that grow much better in a climate where you have more heat. And this, therefore, it's very important now in a very general way to use these technologies, our technology, but also other technologies based on molecular scissors, that we really can address the challenges of the future. With classical breeding, this is just not possible, and we will fail.
Well, Adam, that was interesting. Do you see that this compares to other genetic studies somehow?
Yeah, I mean, over the last years, we have seen just a wealth of gene editing studies using CRISPR-Cas9, these genetic scissors. And at some point, you start to feel like maybe you've seen it all. And then someone comes along and is working not just on editing individual genes, but individual chromosomes. So it's really taking this idea to the next step.
Okay, now that we have seen all our 10 winners and 10 different subjects and topics, do you see a general trend in what we have heard today?
Well, of course, they're incredibly varied, both in technique and in application. But one thing you really notice with all the talks, and that I gained by speaking with all the winners, was that everyone is so eager to get this work out of the lab and to patients or into the field, whatever it may be. And it's really exciting to hear this work isn't just at provisional stages, but it's also often getting pushed to that point where it can start making a difference in our world.
And how was it for you personal to jump between all these different topics and issues?
I mean, it's amazing. As a physicist myself, I don't come from the life sciences or from biology, so it's so exciting to me to hear this work, and just to see this progress often seems almost magical.
Okay, great. So we have met all the 10 winners in this category, Life Sciences. Congratulations to all of you again. It's a great big step you made here in the Falling Walls and the Berlin Science Summit. And we have one more important thing to mention. On Friday comes the moment of decision. The jury will decide who of them will be the breakthrough of the year. Friday is our grand final, where we will celebrate the very best breakthroughs in 2020, and I wish you all good luck. So that was our winner session in this category. Thank you so much, Adam, for joining me. You stay with us. And thank you very much indeed, Maya Makarov, for joining us and contribution. We highly appreciated it. Thank you so much. Have a nice day. Since we will continue immediately with our next winner session in the category Emerging Talents, stay with us. Don't miss the next winner session. And I have something else I would like to recommend, and this is our Q&A session. It will start at 3:15 p.m. CET. It's a wonderful opportunity to get in touch with all our winners. They will be there. They will be talking to you. Just go on the Q&A button and you will be transferred to the chat rooms. They are moderated one-to-one, or you can talk in smaller groups. So it's a great chance to talk on eye level to each other. So thank you very much for watching this. Stay connected, since we will continue with our next winner session. And this is Emerging Talents, a very special category. I'm sure you have something to tell about this as well. Thank you so much. See you later. Hello, everybody. Thank you so much for joining us at the Falling Walls Breakthroughs of the year 2020. Glad that you're with us. My name is Astrid Frohloff. I'm a German TV moderator and journalist. I have the pleasure to guide you through this session. Well, it's an important and highly exciting day. Presidential elections in the United States. It's still not clear who is going to be the winner. But here at the Falling Walls, we do have our winners already. We do have 10 winners in this category, and this category is emerging talents. And we are proud to announce to you some of the best young researchers one can find. So in the studio with me is Dr. Adam Levy. He is my co-moderator. Nice to have you with me, Adam. You are a scientist yourself, a physician, right?
A physicist, yes.
Physicist,
yeah.
Okay. And you have made it to your business to explain, to present, and to communicate science issues to a wider audience. I talk to you later about this. But I would also like to introduce to you our guest who is online connected with us. And this is Claudie Aigneret. She is the distinguished jury chair of this category. In fact, she has been chairing the jury of this category for the past few years. And it's a great pleasure to have you joining us again. Hello.
Hello, Astrid.
Before we will talk a bit about the background of this very special category, let's have a look at the board of the jury members. Each of them is a distinguished experts in his field, and the jury did a great job in discussing intensively about the winners. Here you've got a picture of the jury members. They had a difficult job to really decide who the winners will be in our category, Emerging Talents. So let me ask you, Claudie, it's a privilege to have you joining at the winner session today. You are not only the jury chair, you have been a senior advisor to the European Space Agency Director General. You became the first European woman astronaut candidate. You performed 2 space missions yourself, and you served as Minister for Research and New Technologies and as Minister for European Affairs in the French government. Wow. So let us talk about the special category today. All the participants were qualified in international Falling Walls lab events where they had to pitch their idea or their project. So today's winner have gone through a few selection rounds before the jury voted. Can you say, Claudie, we are looking at global super talents in this category?
Yeah, yes, you are right. It was a fully online process of selection. 1,500 applications and a few selection rounds in 45 labs worldwide to select 98 finalists. And this panel represented a large geographical diversity, a wide range of topics from basic science to concrete application in life sciences, material physics, digital tech, energy, agriculture, and environment. And out of these finalists, a great jury with largely distributed expertise had the difficult task to select 10 winners, and the pleasure to notice the wide gender balance. We have been immersed in an exciting high-level research with young people who are committed, passionate, responsible, and confident in the contribution of this research to provide knowledge and innovative solutions to global or local issues. We had the feeling of meeting the builders of tomorrow. It was a privilege and a stimulating moment with rich and rigorous discussions to present you in a responsible and consensual manner the 10 winners in the Emerging Talent category.
Mm-hmm. In your eyes, what's the value of bringing young researchers, young international scientists together in such a format like this?
When I spoke previously of geographical diversity, I meant both the nationality of the finalists, but also their host laboratory, which are often very far away from their country of origin. Research is an open field of cooperation where laboratory teams allow emerging talent to grow and give them the opportunity to develop their ideas and implement their technical solutions. The projects were very diverse, but always with the ambition of having a major transformative impact through innovation and cutting-edge research. And what they have in common is commitment, passion, perseverance, and collaborative work. Breaking them together is a way to create new links, to generate new ideas through exchanges, to enlarge the network of knowledge, and to become aware of their power and responsibility, and together to break down many walls.
Okay, thanks for the moment, Claudie. I talk to you later. Now I'm excited to meet the top 10 winners in this very specific category, emerging talents. Now let's get a little feeling of how this could look like. Adam, you have had the chance to talk to our emerging talents. What was so special about this category for you?
Well, it's so interesting to meet with people who are at that earlier stage of their career, but still see achieving incredible work. And one thing I really noticed across this category is not just amazing research, but really fantastic communication, which as a science communicator, I really value.
I can imagine. Okay, talk to you later, Adam. Now, we would like to start with our winners, and I'm delighted to announce to you our first winner today, and this is Chelsea Tucker, University of Cape Town, who wants to break the wall of energy insecurity in Africa. Congratulations. And let me ask the jury chair, why did you decide to vote for this project, Claire?
Sub-Saharan Africa is energy scarce in terms of fuel and electricity, hindering economical growth and increasing vulnerability. Chelsea is a passionate and convincing coordinator of an end-to-end waste-to-fuel initiative. In her host lab in South Africa, they designed a decentralized diesel system as a modular carbon-neutral off-grid system based on a robust technology to produce electricity and diesel from organic waste with a major innovation, a brand new catalyst with high activity and stability, taking advantage of an old process, producing a low-quality fuel from coal. They design a way to break the wall of energy insecurity in Africa, a promising Green Deal for Africa.
So then let's learn more about this project.
Breaking the wall of energy insecurity in Africa. Chelsea Tucker, University of Cape Town.
So Africa is incredibly energy scarce. And what this means is that we don't have a lot of electricity or a lot of liquid fuels. So we're trying to find a way to create both electricity and diesel in a decentralized way to reach remote regions of Africa, as well as be equitable for all Africans and, of course, green. So the Waste to Fuel Initiative has developed something called the Decentralized Diesel System. This is a modular off-grid waste-to-fuel process that can provide electricity and diesel in any region regardless of infrastructure, as long as that region has waste. We are wanting to develop a vast network of regional jobs from waste pickers all the way through to energy entrepreneurs and engineers, providing marginalized communities with a way to enhance local economies by the creation of low-carbon energy.
And how do you actually hope this work could influence local communities?
So what we're hoping to take the creation of energy away from behemoth companies across the world and actually bring energy creation back into the hands of communities. So not only are they able to enhance local economies by creating both electricity and fuel, but also provide jobs for a wide range of individuals, both skilled and unskilled labor.
And what stage is this work at today? Is it already out of the lab?
It's almost out of the lab. What we've done is we've developed the entire process across basically 3 different continents and have been working towards developing the various parts of this modularization. Now we're at the stage where we're looking for funding. We want funding to develop the first major prototype and then potentially to start rolling it out into a region of West Africa.
Can you give a sense of just how big the gap is for electricity and for fuel on the African continent?
So Africa is extremely energy scarce when it comes to both electricity and liquid fuels. So from the electricity side, there are areas of West Africa that have extreme power outages, up to 4,600 hours without power. That's 190 full days. So in order to offset this, many governments try to use liquid fuels as a backup electricity generation source. But on that front, Africa only actually refines 2.3% of the world's liquid fuels. So we don't have either in order to actually uplift our own economies.
What ultimately is your dream for the impact this could have on the needs of Africa?
The overarching dream is that instead of large corporations coming into Africa and providing Africa with energy, we would be able to allow Africans to provide energy for Africans. We would be using our own resources, and there would be one of these in every community. So every community would have a small little depot where they drop off their waste, and that waste is turned into diesel, which is then sold back to the community. And all of the people involved here, all of the stakeholders, are locals. So we're wanting to take jobs out of these main cities and back into actually rural and remote regions where they're so desperately needed.
Let's shift directly from South Africa to Israel. I'd like to announce our next winner in the category Emerging Talents, and this is Amit Rimon, master student of the Hebrew University Jerusalem. And the future breakthrough is about antibiotic-resistant infections. So the question for the jury is, what was the reason for you to vote for this project?
The wide use of antibiotics have led us to a point where bacteria are resistant to almost all antibiotics, and we are today powerless to treat these infectious diseases. Amit proposes the use of viruses which specially destroy bacteria. These are the bacteriophage or phages that are genetic elements easy to manipulate or engineer. Amit makes the phage therapy, an old idea, work, introducing a personalized approach in this therapy. Large phage banks, precise matching between phages and targeted diseases, and the jury was impressed by the preliminary clinical results. So a fight for cure between viruses and bacteria with a happy end.
Okay, let's have a look at this project.
Breaking the wall of antibiotic-resistant infections. Amit Rimon, the Hebrew University, Jerusalem.
So we are working on a breakthrough that encounters the problem of antibiotic resistance that is rising worldwide. And our breakthrough is to utilize phages, which are viruses. Surprisingly, not all viruses are bad. And we use these viruses that infect specifically bacteria and do not cause any harm for the human body. And we utilize them in order to treat patients with antibiotic-resistant infections, as we have successfully treated 4 patients already.
Give a sense of just how big a problem antibiotic resistance is.
Today, 17 million people around the world die from antibiotic-resistant infections. And this number will only rise with time and with the widespread usage of antibiotics. So it's a really major problem that we only now are on the edge of.
And I think to many audience members, it might sound strange to actually use a virus to attack bacteria. Is this something that we know to be completely safe?
So this mechanism of attack, the phages are viruses that are specifically attacking bacteria and were found back in 1917 by the Hedrale. And they were long forgotten because of the usage of antibiotics, but they are present and they have been used in basic research, and there are elements that are known to kill bacteria, but simply they were not used in clinical surroundings yet, because the antibiotics were given a proper solution. But right now, antibiotics are not giving us a proper solution, and we must utilize phages, and we must find new tools to add to our toolbox in order to overcome this antibiotic resistant problem. And we are going back to phages that have been found many years ago, but have been long forgotten.
What is your ultimate hope for the impact that phages could have on the treatment of otherwise resistant infections?
We have so far treated 4 patients, and we have great success in most of them. And we believe that phages don't come to replace antibiotics. They come to help them where they are lacking. And actually we have treated phages in combination with antibiotics. And phages have made the antibiotics work better. And our first patient, Hassan, was treated with phages and with antibiotics. And instead of leg amputation, his leg was saved. And today he's healthy and he's walking on his 2 feet at his house. So we believe that it has tremendous effect. It still needs to be understood in what specific infections it is most helpful, but it is a major tool that in a toolbox that we must utilize, and it can really make a difference for every antibiotic resistant patient out there, as we have shown with Hassan and other 3 patients already.
Adam, can you give us a picture of how does this compare to other approaches to tackle this problem?
Well, we've seen antibiotic resistance is a huge problem, and there are many approaches to tackle it. What's interesting about this is it's not developing something completely new. It's harnessing bacteria's natural enemy, these phages, and I think that really sets it apart from other approaches.
Okay, now let's announce our next winner in this category, Emerging Talents. And this is Marie-Claude Bay, Eidgenössische Materialprüfungs- und Forschungsanstalt Dübendorf, this is Switzerland, and she's gonna break the wall in fast charging. Clary, can you say what convinced the jury most about her work?
Marie-Claude is a material scientist, very enthusiastic, convinced on the success of her idea, and convincing, really, in order to break the wall of fast charging of batteries that are more and more adapted at a large scale. She proposes an alternative to lithium-ion batteries by developing a sodium-ion battery based on a sodium alumina ceramic electrolyte and a sodium metal anode, separated by an ultra-clean interface obtained by a patented heat treatment. And with her research, she kills 3 birds with 1 stone: a non-flammable and non-toxic material, an abundant material, and 10 times faster charging.
Okay, so then let's learn more about her work.
Breaking the wall of fast charging. Marie-Claude Bay, EMPA.
So we are working on a new battery technology where we replace the negative electrode of today's lithium-ion batteries, which is graphite, by a metal material, which is sodium. And we also replaced a separator, which is the layer that separates both electrodes, which is a liquid in lithium-ion batteries, with a solid electrolyte. So in my case, it is a sodium alumina electrolyte. And this new battery technology would enable 2 times longer user time, because sodium can store more energy than the graphite material. And it would also enable 2 times faster charging time than lithium-ion batteries. And also this new battery technology will be safe, because we replace this flammable liquid separator with a solid material which cannot burn. And we use abundant and non-toxic materials. So sodium is 400 times more abundant than lithium. And this is really a big step towards a sustainable future.
Now, what's the primary motivation for this? Why not just refine what we already have with lithium-ion batteries?
The idea of using a metal negative electrode would enable longer user time. So this is something that I think would be a big step in the battery field. We could use the battery much longer than what we can do at the moment.
Now, why is battery technology becoming more and more important?
I think battery technology is very important today because we would like to store the renewable energy, for example, from a solar source or wind power, for example. And we need to find a way to store this energy. And one way to do that would be by storing energy in the form of batteries.
What stage is your work currently at, and what would it take to get it out of the lab and into, say, electric cars and laptops?
I think it will take still a long time, so we need to do more development, and I think it will take years, maybe 5 to 10 years, to have the battery ready in the market.
Is your battery technology particularly well suited to any one application?
I think for the beginning, I would like to start for small size applications, for example, for smartwatches or for Bluetooth headsets, for example, before going to a bigger market like electric mobility.
Now, how would you compare the improvements you could see with your technology versus what we might be able to expect just by improving existing lithium-ion technology?
So by improving lithium-ion technology, I think we can improve the energy that they can store by 10% maximum. And with this new battery technology, we can double the energy density. So this is a big step forward.
Well, batteries, of course, is a major issue in science. Adam, how is this particular idea we just heard linked to others?
Well, we will see over the course of Falling Walls this year several winners talking about battery technology, and there's a good reason for that, because batteries are so crucial for the energy future that we have to build going forwards.
Okay, now our next winner. We're going to Mexico, and I'm delighted to announce to you Humberto Martinez Baron. He is a student at Nerité, and he's working on the breaking the wall of unsustainable agriculture. So, Claire, what did the jury like about his work?
Smallholder farming practices will not be able to withstand the necessary growth in production in a sustainable way. Small-scale farmers don't have enough information to grow food efficiently. The waste of fertilizers, the soil depletion, endangers their survival. Humberto has developed a soil nutrient analysis sensor that can help farmers optimize the use of seeds, water, and fertilizers, thus increasing productivity sustainably. It's an ingenious connected system on site, a low-cost infrared spectral analyzer that determines the nutrients present in the soil and give back advices through customized AI and algorithm. I would say that's modernity at the service of rural tradition to make agriculture sustainable, profitable, and maintaining the rural lifestyle.
Okay, and here comes a video introducing this project.
Breaking the wall of unsustainable agriculture. Humberto Martinez Baron, Nerité, Mexico City.
So we realized that today, small farmers are facing a big dilemma. That dilemma is that larger farms have access to better technology, and therefore they can optimize their costs. Smaller farmers who cannot afford these technologies are usually left behind and therefore incurring larger costs, and it also can impact the environment in a very negative way. Specifically, I'm talking about fertilizers. When farmers use fertilizers, they need to know exactly how much of it they should be applying. Too much, and they're wasting money and potentially harming their plants, and too little, and their plants won't grow properly. So the device we have developed is a device that, for a very affordable cost, will help farmers determine how much fertilizer they should be using by sensing the properties of the soil using infrared light.
These kinds of device already exist, but what did you do differently which allows this device that you've made to be so affordable?
We realized that only a few wavelengths of light were actually relevant for the analysis of the soil. So we selected those and used a cheaper electronic component that can be repurposed to immediately sense and detect light at the same time.
Now, is this all about saving farmers money, or are there other impacts of this as well?
The fertilizer that is not used by the soil ends up polluting one of three different environments, either the water, the soil, or the atmosphere. The most dangerous of those is the atmosphere because it turns into nitrous oxide, which can pollute 300 times more than carbon dioxide.
And what stage is this device currently at, and when do you hope to actually start getting it into the hands of farmers?
So right now we are finishing our first prototyping stages, and what we need is to gather a large enough data set so that we can do calibrations specific to each area where we want to release this device. Once we have enough of that data, we can immediately give that device to farmers so that they'll start using it.
Could you give a sense of just how much work it takes to go from this idea to something you have now which is working?
Well, I guess most engineers and technical people will know that the problem with developing something new, and especially when it's an open area of investigation, is that you'll come up with the idea and then you'll find 1,000 reasons why it might not work. And when you start looking into it, then you might find one or two reasons that it might work. And then when you start doing those, then maybe the electronic side and the industrial design side work perfectly separately. But when you put them all together, it falls apart. So you have to go back to the drawing board and start over. And I think that's the most challenging side, that theory is not everything. You have to realize that putting it all into one single device that actually works is really a really hard challenge.
What is the broad impact you hope this device could have on the world?
Well, what I'm hoping to achieve with this kind of technology is that especially small farmers in developing countries, who are generally the ones who waste the most fertilizer, will know how much to apply. That can save them money, but it can also improve our environment, because this, especially the nitrogen that is not used by the soil, ends up contaminating either the soil, the water streams, or the atmosphere.
Let's shift from Mexico City to Nairobi. I'm glad to announce our next emerging talent, Mutambe Kariuki from Fast Egger, Nairobi. His potential breakthrough is dealing with artificial intelligence. Claire, what is the reason for the jury to vote for this project?
We met the man who wants to build the African AI. He creates a startup, Fast Egger, to build appropriate labeling tools for the Africans' low internet and low computing power context. They are labeling African-focused satellites images for AI use cases, and they are developing artificial intelligence, which they will provide to companies via API. That means AI as a service. And furthermore, they train African youth to do the labeling of the dataset. Overall, he wants to build African AI to use the power of AI to speed up sustainable development for Africa by Africa.
Okay, so let's have a look at this project and learn more about it.
Breaking the wall of democratizing artificial intelligence. Mutambe Kariuki, Fast Egger, Nairobi.
We are building the labeled datasets that will be the learning tools for artificial intelligence. And we're particularly focused on the African continent, because just like you need to teach a child based on the context they are in, there is a huge lack of contextual learning data. This is training labeled datasets for the African continent. And this is the big gap that we're seeking to bridge by democratizing artificial intelligence here.
Now, these labeled data sets are the tools through which artificial intelligence can learn. Why is it important that we have labeled data sets coming out of the African continent?
It's extremely important. I think recently, about a month ago, there was an AI that was asked to clean up some images, and it was given an image of the former US President Barack Obama, but because it had been fed on mostly images from people of European descent, it ended up cleaning up the image and coming up with a face of someone of European descent, even though it was fed with an image of Barack Obama, who's clearly of African descent.
And so far, what have you actually done to start creating labeled data sets for artificial intelligence?
So far, we have been able to use open source tools and create our own tools that can be used for the space here in Africa, which is low internet, low latency, and we've been able to work on satellite imagery. We went through a program with Airbus. They have an accelerator program focused on Africa. And so we got a lot of competencies around, working with satellite imagery and other Earth observation data, such as drone imagery. And we've started focusing on the area of infrastructure, especially solar. We engaged with a potential client who's a solar mini grid developer, and they needed the algorithms that can be developed from this labeled data set to be able to identify areas where they can put solar panels. We know that about 600 million people on the African continent do not have access to electricity. And one of the ways that this gap can be bridged is through solar panels. And, having this machine learning algorithm being able to identify the places would really help them do this faster.
What's your hope for what artificial intelligence can bring to Africa if you do have these labeled data sets?
I have great hopes. Everybody talks about AI as being the new electricity, and particularly I see it as an opportunity to achieve the sustainability development goals on the African continent much, much faster. We are not going to be able to create enough doctors and medical officers to satisfy the population growth that we're having. We are not going to have enough technicians. We're not going to be able to have enough educators to increase the literacy levels. And AI can be the one to come in, to bridge these gaps and enable the continent to reach their sustainability development goals much, much faster.
Adam, I'm wondering, how does this address concerns about artificial intelligence?
Well, we'll hear, and we have heard at Falling Walls, concerns that artificial intelligence could entrench inequalities in our society. And it's really refreshing to hear a project which tries to take the promise of artificial intelligence and apply it to a use case to the African continent, where normally it isn't applied.
Mm-hmm. Okay, thank you. I'm glad to announce our next winner now in this category, Emerging Talent. It is Shawana Tabassum, University of Texas at Tyler, and the project title is Breaking the Wall of Neonatal Health Disparity. So, Claudie, what is the explanation of the jury?
Shawana presented a device in order to monitor biomarkers of sepsis, jaundice, and hypoglycemia on neonates, babies, at the point of care in rural areas of less developed countries for early diagnosis and treatment. The jury was seduced by the performance of the proposed device to break down the wall of neonatal health disparity with a reliable, sensitive, rapid, low-cost, miniaturized, point of care, easy-to-use device that can impact immediately and concretely neonatal health care. Shawana is a bright engineer in the field of microfluidics and optics. She is a true problem solver, and furthermore, she is strongly dedicated to education and training. Undoubtedly, she is a great ambassador for women in sciences and engineering.
Thank you. Then let's have a look at the presentation of this project.
neonatal health disparity. Shawana Tabassum, University of Texas at Tyler.
I'm breaking the wall of neonatal health disparity by developing a rapid, cost-effective, and point-of-care diagnostic solution for infants. So specifically, the device is for monitoring sepsis, jaundice, and hyperglycemia. So sepsis, it alone causes 3 million deaths worldwide, 3 million deaths of kids. And our device provides rapid detection in less than 10 minutes. Also, because it provides rapid detection, it can accelerate the treatment substantially and save millions of lives. At the same time, it provides bedside monitoring and will be an affordable solution to the poor people. So these are the features which are missing in the current technology. So that's why through this device, we hope to attain healthcare for all in the long run, because every infant, no matter where it is born, has the right to live.
So what were you actually able to do in order to make this test more affordable, more portable?
We are using some cheaper materials, which can be like the overall package will be much less costly. And as I said, like still the device might not be affordable to those people who live below the poverty line and cannot afford 3 meals a day. But the thing is, we are hoping to partner with the UNICEF, local NGOs who are working for the betterment of these people. Like they are providing free diagnostic devices to the communities as a whole rather than to each family, because it will be difficult for these families to afford or to buy such devices for their own family, rather than if we can provide 1 or 2, for example, to a small community, then it will be all the people in that community will get access to the device. And this is what the UNICEF and local NGOs are already working on it.
And on a personal level, what actually inspired you to work on this?
When my son was born, so he is now 1.5 years. So back when he was just born, he was a preterm baby and he had hypoglycemia at birth. So at that time, like I was really, I could really feel the pain of those mothers, especially in my country, where they cannot afford the basic healthcare for their kids, and they have to just watch their kids dying because they just cannot afford the healthcare for them. And when I was back in my country, although I have seen this scenario with my own eyes, their pain could not really reach my heart until I went through the similar kind of pain here in the US. But the major difference here was that I was really privileged because I was here in the US when my son was born. So I could afford all the healthcare for him. But how about those mothers living in those rural communities? Like they cannot just afford the healthcare and they have to watch their kids dying. So that incident really moved me to an extent that at that moment I just decided and made up my mind that I'm going to use my research for the betterment of the society, for doing good to those poor people and bring some smile to their mother's face. And that's when I embarked into this mission of ensuring healthcare for all by breaking the wall of neonatal health disparity.
So we are presenting the top 10 winners in the category Emerging Talents, but only 1 out of 10 will make it for the grand final on Monday. On the 9th of November, we will celebrate the best of the best and the breakthroughs of the year 2020. The jury has to decide. I'm excited to see who will make it. So let's introduce our next winner, and I'm glad to announce Tanmay Dutta from Switzerland. The project title is Breaking the Wall of Data Latency and Loss. So what was the reason for the jury to go for this project?
Tanmay takes us into the complex world of skirmions to break the vicious circle that causes latency in computer processing and data loss in storage. Breaks this vicious circle by introducing stable, nano-sized skirmions in the form of bits in a ferrimagnetic material arranged in a unique structure. And the jury was impressed by his conviction and pedagogy in explaining a cutting-edge research that promised a major improvement in the current performance of data storage and processing, which will enable the potential of digital technologies and apps to be exploited to the fullest extent possible.
Thank you, Claudie. So let's have a look. Here comes the video.
breaking the wall of data latency and loss. Tanmay Dutta, EMPA.
Today, data is the oil of the century, or the gold of the century, let's say. And being the most valuable thing, it is important to find solutions to store all the numerous amount of data we are generating every day. And we won't believe we are generating 3.5 quantabytes of data every single day, and it's too huge to store in our limited capacity. That too with less energy and also at a less energy cost. So my solution is to find or generate fermions which are stable, energy efficient, and can be moved around much efficiently in ferrimagnetic materials. And here it is important that ferrimagnetic materials are the most promising candidates to hold these fermions, which are basically bits of information, which are 10 to 20 nanometer wide, so they're really small. And this is my breakthrough to make this really possible, where we can store information in the smallest, densest, and fastest possible form.
Can you give a sense of what fermions are and how they allow us to store data so much more densely than conventional hard drives?
See, conventional hard drives are mostly used based on domains, magnetic domains, which are either north or south oriented. Now, they can be small, but there is a physical limit to how small they can be. So because of the laws of nature, they cannot be as small as a fermion can be, for example. And that's why fermions are more important, or are for our memory for the future, because they can be very small as compared to a domain, which is basically what you find in hard disk drives. And at the same time, they are also energy efficient, and they can be very densely packed, giving us a very high density information storage.
Do you have a sense of when we might actually see hard drives which are made with these novel fermions?
Well, that's a very difficult question at this moment, because I would say that industry has, as far as my knowledge goes, industry has not yet started making fermionic devices even on ferromagnets, let's say. And I am working on ferrimagnets, which again is a new class of material, and will take some time to be studied for the different errors and problems it can have, the optimization process. But yes, given the amount of data storage requirement we are facing today, I would say that we'll have to all scale up our efforts, the industry and the research groups around the world. And I think maybe if everything goes well, maybe a decade or so, and we might even have a device. And probably that's me being too optimistic, at this moment. I have to admit. But then that's the way. I mean, we researchers have to be optimistic about it. Otherwise, we can't go ahead. That's the only way.
Adam, what could be the impact of this innovation for science? What do you think?
Well, I think for many of us, myself included, we can sometimes think of data as ethereal, not in any physical place, but work like this really reminds us that it does have to exist somewhere, and we need to make room for it. And it's great to know that some researchers are planning for that future.
That's right. Okay. So let's switch now to a health issue. Our next winner is Khalil Musi. From the King Abdullah University of Science and Technology, Tuwal. The project title is Breaking the Wall of Coronary Artery Diseases. And I'm asking Claudie, what is the reason for the jury to vote for him?
Khalil proposes an innovative alternative to surgery or stent placement in case of narrowing of blood vessels due to arteriosclerosis, the so frequent coronary artery disease. He develops a 3D printed hollow microneedles integrated in a balloon catheter for localized and targeted delivery of therapeutics to reduce or eliminate non-invasively the narrowing of blood vessels. Associating the engineer and the doctor, here is a precision but low-cost engineering for safe, non-invasive, personalized solution for a frequent and increasing pathology.
Thank you. So let's watch a video to learn more.
Breaking the wall of coronary artery disease. Khalil Musi, King Abdullah University of Science and Technology, Tuwal.
So the project that I'm working on
in a team of researchers, engineers, and cardiovascular surgeons is the development of a microneedle balloon catheter for vascular drug delivery, especially for heart disease. Heart arteries, they get narrowed due to an accumulation of cholesterol known as plaque, and then this will reduce the blood flow to the heart muscles and can lead to heart attack. We have developed a microneedle balloon catheter that will allow doctors to inject medicines directly to the cholesterol layer in order to control and reduce the narrowing. We are using a high resolution 3D printing technique to fabricate biocompatible and highly customizable microneedles. We have done animal experiments, and we have demonstrated a precise delivery to the vessel wall. Our solution does not only save lives, but it can revolutionize the way vascular drug delivery is being done today.
What work still needs to be done before this can get out of the lab, out of experiments, and actually start helping and saving patients' lives?
So, this is like a device for patients. So we need to go through multiple steps. Now we started animal experiments. We have done, let's say, ex vivo or in vitro experiments. Our next step will be to move to live animal experiments. Then, once we optimize and figure out and tackle all the challenges and make sure that our device is ready to the next step, then we can move further to human trials. So there are like a couple of steps just before the human trials.
Can you paint a picture of what this device actually looks like, kind of what it's doing inside the body?
So imagine that you have a balloon catheter. So this catheter is a tiny tube that can be guided inside the blood vessels. All right, and now we have a balloon on top of this tiny tube. On top of this balloon, we have placed microneedles. These microneedles are now protected inside the balloon folds. Now we can guide safely this balloon catheter. Once we reach the narrowing, we can inflate the balloon. While inflating the balloon, the microneedles are now open and they are directed to the narrowing. With the inflation of the balloon, then the needles are pushed inside the blood vessel directly to the plaque. And after that, we can inject basically a therapeutic agent or medicines. After that, we deflate the balloon. The microneedles are safely inside, go back safely or retrieve it safely inside the balloon, and we can again retrieve the catheter from the blood vessels.
In our category Emerging Talents, we've got two more winners, one from Argentina, one from Japan, and I'm delighted to announce now Micaela Belmonte, National Technology University, Buenos Aires. She is aiming for breaking the wall of safe water access. Claudie, what was most convincing for you in nominating her?
We were scared by these figures. According to the WHO, 200 million people are affected by arsenic and fluoride presence in groundwater. Using and drinking contaminated water during a long period of time could cause diseases, educational difficulties, and inequality to get out of poverty. Micaela proposes a twofold solution: arsenic and fluoride removal by adsorption and co-precipitation using a reactive filter based on iron and sand as a low-cost support, and cementing the waste generated for safe disposal as a building material. A simple idea to clean and build the future of millions of people.
So let's meet this emerging talent. Here's a video.
Breaking the wall of safe water access. Micaela Belmonte, National Technological University, Buenos Aires.
So imagine that every time you turn on the tap,
the water you were drinking or using for cooking or just for recreative activities was highly toxic and would produce several diseases to you and your lovely ones. Imagine that it can cost you your life. So what if I tell you that this is the situation that 200 million people deal with because of arsenic and fluoride presence in the groundwater? What if, apart from that, most of that population live in extremely poverty and with no access to education, being unable to be conscious of the situation? It is not only about health, it is also about inclusion and possibilities of better life conditions.
Now, water filters exist already. What makes what you've created different?
First of all, our filter is pretty much economical. It can also bring down higher concentrations of arsenic and fluoride, including all the zones with very high concentrations. It can also abate the most toxic and difficult to remove type of arsenic. And we also think about the treatment of the waste, which is something that hasn't been done before.
Now, can you give a sense of just how widespread a problem unsafe drinking water is?
Drinking safe water is a human right, and we should all be able to reach it. So actually, there are 200 million people affected for this situation. It's an issue that affects people from all around the world. And many of that population live in extremely poverty. So they can't afford a commercial solution. They are also not noticed about how important it is and the consequences that it can bring to their life. There are many diseases related to this situation, to this issue, such as cancer, bones deformation, tooth decay, gangrene, and also mortality.
Now, of course, developing this is just one step. How do you then make sure it gets out there and into the hands of the people who need it the most?
As it is a problem that affects the most vulnerable ones too, and many of the population is in poverty, as I said, we need the contribution of state and public organizations and nonprofit that can help us to ensure the population to reach the product. It is necessary a mix up of many organizations. We have an efficient solution to this issue, a solution at everyone's reach and with no environmental impact.
Adam, have you been surprised to learn about all these details of this problem?
Yeah, so while it's a problem I'd heard of before, understanding that it's hundreds of millions of people affected is just incredibly distressing, but it's very reassuring to know that it's a problem not only being worked on, but being worked on in a way that will hopefully be affordable for the millions of people affected.
Yes, that's true. Well, we are approaching now our last winner for today, and I'm delighted to announce Jonas K.H. Fischer from the University of Tokyo. He is aiming for breaking the wall of energy-efficient data storage. And I'm asking Claudie, what was decisive for the jury to vote for this project?
Jonas is a bright engineer carrying out fundamental research on the properties of special magnets, the multiferrics, in order to improve the magnetic data storage energy efficiency. Multiferrics are materials that combine magnetic and electric properties, combining the advantage of a resilient magnetic data storage with the energy efficiency of electrical fields. That means no wasted heat and a promise to break the wall of energy-efficient data storage for the next generation of computers and even unsought new devices by the magics of improving those magnetoelectric materials. That's a good example of a basic, deep research to preserve the future of the digital era.
Okay, let's watch the video. This is our last emerging talent for today.
breaking the wall of energy-efficient data storage. Jonas K.H. Fisher, University of Tokyo.
So we are working on so-called multiferroics. I like to call them kind of special magnets. What their specialty is that they can be controlled with an electric field instead of a magnetic field. And the thing why that's great is because electric fields are much more energy efficient than magnetic fields.
And why do we want more energy efficient magnets like this?
So the main usage of these, how we apply a magnetic field and why we want to have a magnet pointing in a specific way is in data storage in our computers mainly and in our internet servers. And if we could make computers and internet servers more energy efficient, I think it would be a great benefit, because if you just think about recent years, we use the internet for everything. So we have a lot more computers and more servers, and if we can make them more energy efficient, we save a lot of electricity.
Now, you're not the first person to think, oh, multiferroics, they could be useful for data storage. What exactly is your project working on?
So yeah, multiferroics is a very wide topic. Our specific multiferroics that we are looking at, that I'm looking at, are so-called conical spins. So the little magnets inside are kind of rotating. It's a very complicated structure that leads to them being multiferroic. And they have a very strong coupling between the magnetism and the electric field. So basically, the only thing they cannot do is do it at room temperature, which is, of course, necessary for us to use it in computers. But we hope that if we understand them better, we can find a material that can be used at room temperatures and then really be used in our computers.
Can you give a sense of just how big the energy use of data storage is today?
So I don't know the number by heart, but if you just think about how many computers we have, how many internet servers, how much this has increased, then reducing the amount of electricity they use would be a huge amount. Like, I'm talking about millions of kilowatt. And just the amount you can reduce it by using electric fields instead of magnetic fields, that would be 10,000 times more efficient. That's the number that has been flowing around.
And is this something that we could potentially see in our computers soon, or is this still very much at the basic principles stage?
So our research is basic research. We are looking at multiferroics with this strong coupling between electric and magnetic fields, but at very low temperatures. So these are not anywhere close to application. There are other multiferroics that can be used at room temperature. Unfortunately, there the coupling between the magnetism and the electric field is not so strong, but they are getting very close to application. Hopefully we will get it in the next few years.
Of course, efficiency is just one of many factors that makes data storage important. Would multiferroics be able to store as much data as we are able to now on conventional hard disks?
Oh, that's great that you asked that, because actually multiferroics could have double the data capacity, because not only can you save on the little magnets, you can also save on, with the electric field, you can control kind of electric order, and on those you can also save data. So you would double the data capacity.
And what is your hope for how multiferroics could change computing in the future?
One thing is, of course, make the computers more energy efficient, which is basically just necessary for us to keep the standard that we have without wasting huge amounts of electricity. But other things that multiferroics can do is we could have tiny sensors for magnetic fields. We could have tiny engines driven by external magnetic fields. So this is the reverse effect of what we want to use for data storage. We want to control magnets with electric field, and the reverse effect could also find many different applications, completely new applications, and not only make computers better.
So that was it for today in our category Emerging Talents. You have met our top 10 winners in these categories, and you learned about their stunning projects. Adam, actually, we have this year of crisis. We have the pandemic. We have climate change, you know, a lot of global problems. Did you actually get some hope talking to all these young people, our talents of the future, and see what they brought and what they're bringing up?
It is really wonderful to hear this incredible work from people still often really near the start of their careers. And while it's important that we bear in mind that this is early stage work, a lot of it will stay in the lab, it is so reassuring to see people who are so passionate, so dedicated, and so talented facing these problems.
Yeah, so we have met all these 10 winners, but it is getting more exciting next Monday, the 9th of November, which is the anniversary of the falling of the wall in Berlin. We will celebrate the breakthroughs of the year. The very best will be celebrated. The jury will choose 10 winners out of 10 categories. This will be our main event during the summit. And at this moment, I would like to thank Claudie Egnery very much for joining us today. We appreciate very much that you have been with us. Thanks a lot and have a nice day. And also, Adam, I would like to thank you for supporting me and joining me here in the studio. You will be with me tomorrow, because tomorrow we will continue our winner sessions, and we will have the categories Social Sciences and Humanities. This will be at 1:15 p.m. German time. And then we'll continue at 2:15, Science and Innovation Management. This is the other category, and we have great winners again to present to you. So something else you should not miss is our Q&A session, which starts right now after this session. You have the chance to talk to all our winners. These are moderated sessions, either one-to-one talks or talks in small groups, and I can highly recommend this. So thanks for your attention. Have a nice afternoon and see you tomorrow. Bye.